A central property of synaptic connections in the nervous system is their ability to change, both during development and as a result of certain patterns of electrical activity. And this is believed to be crucial to modulate the activity of neuronal networks. At the cellular level, a key underlying process is the transport of messenger RNAs and their localized translation. In these mechanisms, RNAs that are maintained translationally silent are packaged into granules, which are transported to synaptic sites where they become localized. Upon plasticity-inducing stimuli, these RNAs are translated into proteins that contribute to changes in synaptic function. So an important question has been how these mRNAs are precisely directed to synaptic sites undergoing dynamic changes. So to examine this problem, we have been using the Drosophila neuromuscular junction, a powerful genetic model system that has allowed the discovery of fundamental biological principles that are conserved across evolution.
An interesting feature of the larval NMJ is that it is highly plastic, making it a convenient and accessible model to study synaptic development and plasticity. As the larva develops from first instar to third instar, before pupariation and metamorphosis into an adult, there is a 100-fold increase in muscle size, and in order to maintain synaptic efficacy, the presynaptic arbor continuously extends by generating new synaptic boutons. In this process, a presynaptic bouton is formed and then recruits postsynaptic proteins that form the postsynaptic apparatus. Very interestingly, we have previously demonstrated that the assembly of the postsynaptic machinery may involve localized RNA translation. So the question is, how do synaptically localized RNAs travel from the nucleus to synapses? A known feature in this process is the involvement of cellular cytoskeleton and associated proteins. Therefore, we investigated the role of a protein that links the nucleus skeleton to the cytoplasmic cytoskeleton, which is Nesprin1. Nesprin1 is a giant transmembrane protein present at the outer nuclear membrane belonging to the spectrin superfamily of proteins. We found that Nesprin1 forms striated F-actin-based filaments that we dubbed railroad tracks that span all the way from the nucleus to the NMJ. Interestingly, these railroad tracks specifically wrap around immature boutons prior to the recruitment of the postsynaptic apparatus. In the absence of Nesprin1, mRNAs that normally localize at postsynaptic sites are missing, and instead they now accumulate around the nucleus, thus suggesting that the railroad tracks serve to direct RNA to new synaptic sites.
What was particularly exciting was that in the Nesprin1 mutants, the NMJ did not expand normally and had far fewer synaptic boutons. This indicates that the Nesprin1 railroad tracks are likely to serve as a path to target specific RNAs from the nucleus to postsynaptic sites. In the absence of these tracks, the RNA failed to localize to the postsynaptic region, resulting in an accumulation of immature boutons, lacking postsynaptic proteins. We also found that the Nesprin1 railroad track association with immature boutons depended upon an unconventional myosin.
Our studies suggest an important mechanism by which RNAs are transported specifically to sites of postsynaptic maturation. In particular, we have uncovered a novel acto-Nesprin1 filamentous pathway, the Nesprin railroad tracks, which serve as a long-range pathway for mRNA localization and synapse maturation. This is of significant importance since Nesprin1 is associated with a variety of musculoskeletal disorders, such as X-linked Emery-Dreifuss muscular dystrophy, cerebellar ataxia, bipolar disorder, and it is also a risk gene for schizophrenia and autism.